All right, so now that we're about 5 minutes in, let's go ahead and get started. Did anybody run into any issues installing any of this? Okay, you can double check by running these load commands. If any of these fails, then that's probably not a good sign. So make sure, like, if any of these fail, then make sure to say in the chat, and hopefully someone will be able to help you get those packages. Okay, so essentially in this session, we're going to be talking about how do you actually use the techniques that we've been teaching you in order to address a biological question. Because ultimately, bioinformatics, it doesn't matter if we can do fancy, computational techniques if we're not able to get back to the biology and address genuine biological questions. And so I guess I should say, because I keep forgetting to do this, that this workshop and this lecture is brought to you by Big Club. And if you're interested at all in coming to our meeting, we actually have a meeting right after this workshop. So stick around and we'll invite you to that. So actually, I think Paul, probably who's in the chat, can send you the invite. Okay, so we've got our libraries loaded now. And so we're going to think about a question to ask, because it's probably easier to learn this stuff if you've got a goal in mind. So I study a pediatric bone cancer called Ewing sarcoma. It's got a relatively good prognosis as long as you catch it early and there aren't a lot of metastases, but it can actually develop drug resistance, and it's one of the most likely pediatric cancers to develop drug resistance. It's actually a really interesting cancer too, because it's driven by one genomic event, and that event is the fusion of the EWSR1 gene to the FLI1 gene. So it's driven by a gene fusion. So a lot of people have been coming up with ways to knock down this gene fusion and to design drugs that can prevent this gene fusion from activating. And the point is that if the cancer is driven solely by the gene fusion, then if you can knock it down and if you can block it, then you should be able to stop the cancer in its tracks. But the question is kind of bigger than that, because it's like, well, will that actually stop the cancer, or are there going to be unintended consequences? And it's actually a pertinent question right now, because there's a drug that just went into clinical trials that is designed to do exactly that. So I wanted to ask this question. I wanted to ask, well, what is the actual biological impact of knocking down EWSFLI1 in Ewing sarcoma? And how does it change the behavior of these tumor cells? So the way we're going to do this is we're going to start by searching for an RNAseq data set that actually has an EWSFLI1 knockdown condition in it. So to do that, I use this abstract search function. And actually, this is the same as if you came over here to recount. If you'll recall, recount 2 is the standardized repository where they have a lot of RNA-seq data publicly available here. So if I come over here and I search Ewing sarcoma in this database, then basically I find all of the studies related to that, and then I can download the data by clicking one of these links. But I don't really want to leave R. I'm kind of lazy, so I want to do it all here. So this is going to be the equivalent of that. Okay, so the first thing I do is that I get the data sets that have this Ewing sarcoma mention in them. So I'll view that here. And I want to find one where someone has knocked down the fusion gene. And actually, it's great. The first, in the third one, I can already find that this is one of those studies. So this is a study where they've clearly been trying to determine what happens when you knock down EWSFLI1 in this cell line. So, okay, great. So I take this one and I can come over here and I can say, all right, that's the study I want. And now Recount also has kind of a convenient function called download study, where it's actually going to download all the data from their database for that study. So we download it, and it's actually, I'm in my downloads folder. So if I come over here to my downloads folder, then yeah, I can actually see that that folder is right here. You might not be in your downloads folder. No matter where you are, though, this line should work, which is basically it's just going to load the RSE gene.rdata object into your workspace. So we go ahead and do that. And now we've got RSE gene. So can anyone tell me what is the class of this RSE gene object? Does anyone remember how to do that? Feel free to put it in the chat. So has anyone figured out what the class is of this RSE gene object? I'll give you another minute if you want. Okay, great. So Jesse got it. Range summarized experiment and summarized experiments. Perfect. So does anybody have a guess as to why it shows? So when you type class of RSE gene, right? It says range summarized experiment and summarized experiment. Can anyone tell me what this means? Why is there not one line, but there's two here? And I'll give you a hint that where it says ATTR package, that's actually it telling you what this part is. Does anybody have an idea of what that's saying? So Ayla asked, is there some part that needs to be unpacked? No, it's already been unpacked. I guess maybe I'm misunderstanding what your question is. So when we talk about in R, when we talk about classes, we can talk about ones that already exist in base R, right? So for example, if I make a list, if I say my list, so I've made a list here. If I look at the class of my list, actually that might not work. Let me see here. Oh, it does. Okay. So it says it's a list and there's nothing else that goes along with it. But not all the classes you can use in R actually are there from the beginning. A lot of them come from packages that you have to download and then load into R. So this class, range summarized experiment, actually is not native to R. It was developed as part of a package called summarized experiment. So that class actually comes from this package. And that's basically what this is telling you here. And whenever we talk about like DE-seq data sets, whenever we talk about things like that, those are also based on the same range summarized experiment from the summarized experiment package. So can anyone tell me, does anyone recall what the structure of a summarized experiment is regarding how the data and the metadata is stored? So just to recap here. When we talk about a summarized experiment, essentially, it's got three main components, which are the row ranges, the assay, and the call data. So the assay is the actual count matrix. So we're talking about RNA-seq, so you've got a count matrix of gene counts and within samples. So we can actually get that count matrix by running this line here with the assay command. So if we take a look at that, we can see this here. We can see on the rows, we've got all of our genes. And in the columns, we've got our samples. So this sample had 32,000 something counts of this gene. And then the call data is the metadata. So let's take a look at that. So this is actually the same samples that were the column names here. They're actually the same samples in the exact same order. And they're the row names for the call data. And now we've got metadata that's describing this. So we know what experiment accession it is. We know how many read counts there are. We know a lot of stuff. One thing we don't know, though, is we don't know which sample is which treatment condition. Which ones have the fusion gene knocked down and which ones are untreated controls or scramble treated controls. So the way we can actually address that, unfortunately, it's a little bit manual, but the way you can address that is by going to SRA, so going to the SRA database, and then actually searching for the project ID that you used to get the data. And then now you can see it here. So you can see these are our accessions. So each of these is one of our samples. And then we can see in the titles, we can see which ones are the knockdown and then which ones are the controls. Let's say that we wanted to make sure we had them in the right order. So we're looking here, we can see that this goes 92 through 98. And then here it goes 92 through 98 from bottom to top. So we know then that the first three in our data frame should be controls, and then the next four should be EWSFLI1 knockdowns. And now when we come over here, we can see that that's already kind of been done for us. Well, I guess I did it, but you're going to have 3 that are SH control and 4 that are SH EWSFLI1. And just as a reminder, the repeat command rep just repeats something X number of times. So 3 times SH control, 4 times SHEF1. And we're going to assign that to the condition column in our object. And so now if we look at our call data again, we can see here that now we've got a condition column that includes the conditions on which we want to calculate differential gene expression. So now that everything's been wrangled nicely, all we have to do is make our DE-seq data set and do our analysis. So we give it the RSE gene. So that's kind of like how we did it before. And then we're saying the design. So the design is what column do we want to use in order to split our data set and calculate differential gene expression. So we want to split it based on condition. We want to compare this condition to this condition. So that's why we're giving it this design. So we go ahead and make this. You can actually just ignore that error message or warning message. It doesn't actually apply here. So the first thing we need to do whenever we're looking at our looking at a new DE-seq data set is we need to look at the Pca analysis. And we can do that by getting the rlog transform of the count data. So this takes just a minute. And then we can do this plot Pca command to actually look at the Pca transformation of that. And I'll zoom in here so you can kind of see what's going on. But Basically, from this analysis, we can see that almost all the variance in the data occurs on this first principal component, and that the first principal component really nicely splits our two biological groups, so our treatment and our control. This is great. This is kind of what you really hope to see when you're doing a differential gene expression analysis. This means that the technical variations, so things such as batch effects and mistakes that you made and contamination, is not nearly as strong as your biological variation, the thing that you actually care about, the difference between your treatment and your control. So now that we like our data and we think it's, we think it's good enough quality to analyze, we go ahead and we analyze it. And that just takes a minute. Okay. And then what we're going to do is we're going to get our results. So you might notice that I'm actually specifying the contrast argument here. And the reason is because when we do differential gene expression, terms like overexpressed and underexpressed are actually completely relative. It's based off of what was the numerator and what was the denominator when you did your fold change, when you calculated like expression fold change. So here we're actually explicitly saying I want to compare I want SHEF1 to be the numerator and then SH control to be the denominator. So I'm saying when I get the fold change, it's the fold change in expression of treatment over control. And then when you give the contrast argument, you first tell it what column are you making that distinction based on. So it's a condition column, and then I'm saying SHEF1 divided by SH control is going to be my fold change. Okay. So one of the first things you're going to want to do to just make sure the quality of your results are makes them trustworthy, you are probably going to want to do an MA plot. So we mentioned this last time. Simple enough to do this in DESeq because it's got a built-in function. So MA plots, what is this telling me? Well, this is telling me how does my fold change relate to the degree to which each gene was expressed at all in my data set. So I actually see kind of a disturbing trend here, which is that there's a bunch of genes on this side and on this side that have pretty low counts. So they're not expressed very highly, but they have a huge fold change. This is actually one of the main technical hurdles that RNA-seq has to kind of overcome, which is that the variance at low levels of expression can be extreme. And that's why you can get sort of extreme fold changes even if you didn't have a lot of expression. Now, these things should not be considered significant, and they're not blue, so they weren't called as significant. But still, when we want to visualize our data, we want to use log fold change. So it can be really useful to get a more accurate log fold change. And there's actually a really nice kind of new way to do that, which is to use the log fold change shrink function from DESeq. And basically what this is going to do is it's actually going to find those instances where the fold change was exaggerated. And it's going to basically use a statistical model to shrink them back to a more appropriate place. So this would be what we would want to run to do that. So it's giving you kind of a warning message here. It's telling you you're using the normal model for log fold change shrinkage. It's telling you what paper it came from. It's also telling you that the normal model isn't as good as APE GLM or Asher. To be honest with you, this is really just for visualization. So I personally think that it's kind of up to you which of these you choose. I'm going to pick the one. In this case, I picked normal. I'm going to give the one that seems to make the most reasonable transformation of the data. So if you want to try the other ones, I highly recommend it, just so you can see why I might have picked the normal model in this case. All right, and so that should just take a second. maybe a little bit longer. Okay. I swear this was faster earlier for some reason. So Aola asks, do you only want to take the log fold change when you see those values in the plot MA? So are you asking, do you want to use the log fold change when you see, I guess I'm not sure what your question is. Hopefully someone can. So, okay. So maybe Simon, can you, well, I mean, we're actually kind of ahead of time at the moment. Oh, okay. Gotcha. So yeah, so basically the coefficient argument, that's your way of telling LFC shrink what contrast you are making. So if I look at results names of DDS, it's going to tell me what results are available in this object. So it says intercept. I genuinely don't even know what would happen if you tried to get the intercept result. I'm pretty sure it would not tell you anything informative. What we care about is this condition SHEF1 versus SH control. That's the main contrast we care about. And that's the second of the two. So we say coefficient equals 2. So hopefully that helps. Aola, I think I might not have gotten to your question, but hopefully this is kind of making some sense. Basically, when we do this and we take a look here at our plot MA, we can see how it's changed. Now, so basically before, those sort of spurious genes were like all the way over here, and they got shrunk all the way back to the midline. And now we see a more normal, natural distribution of log fold change with respect to normalized count mean. So this is, I would say this is superior to what we had before regarding how we might want to use log fold change in order to visualize the data. Okay, so let's take a look at our actual results here. And actually, I'm going to get rid of this so it doesn't mess with anything. All right, so we can see here for every gene, we've got our differential gene expression stats. And the ones we really care about tend to be the log fold change and the p-adjusted value. If we sort based on p-adjusted value, we can see the genes which have the strongest differential expression between our conditions. And you'll notice two things. The first thing you might notice is that our p-adjusted value says it's 0. It's not actually 0, and we'll actually have to do something to deal with that later. But for right now, just know that the number is actually so small, R is not capable of displaying it. And so it just rounds it to 0. The other thing is that our genes are not very aesthetically pleasing, and it's really hard to even tell what this gene is without searching it. And you don't really want to find yourself searching one gene at a time all the way through your data in order to interpret it. So the first thing we do is we come over here and we use the Ensembl database to convert our gene IDs to gene symbols. So I had you download and load this database. And so we're just going to take a quick look at what's in here. So if we just take this and we print it to the screen, it tells a lot of information. So first, it tells us that it's an SQLite database. It's a type of SQL-based database. And it also tells you information about when it was created. And it tells you there are over 63,000 genes and 216,000 transcripts in the database. So to first get an idea of how to get this mapping of IDs to symbols, it's useful to know what are the columns of the database. So like if you think about it like a data frame or like an Excel spreadsheet, the rows might be your genes and the columns might be data describing those genes. So let's look at our columns. So we can see we've got different kinds of gene IDs. So Entrez ID is a type of gene ID. We can see other information about the genes. So for example, we can see what chromosome is it on, what strand is it on. We can see how long is that gene. And there's actually quite a bit of useful information here, even beyond what I'm going to show you today. But the main thing we care about is mapping the gene IDs, which are our Ensembl IDs, to gene symbols. And so the way we can do that is we can select from this database, and that's what the statement's saying here, selecting from this database the symbols. And we're going to do that based on the keys. So when we talk about a database, when we say keys, we really just mean index or row names. It's actually quite comparable. So let's take a look at the keys. Now the keys are Ensembl gene IDs. So that's great. We have Ensembl gene IDs here. And so the keys of the database are already what we need to convert from. So all we have to do is select from the database based on those keys, and then say, what do we want to convert to? So we want to convert to symbols. So let's take a look now at this. And we can see we've actually done this. We've returned kind of a nice mapping between gene IDs and gene symbols with that command. Okay. So what do we need to do now? Well, we need to, for each one of these, we need to find its equivalent in here and then map it to a symbol. But there's actually a really efficient way we can do that. All we would have to really do is join these two data frames based on the gene IDs. And if you'll recall from our tidyverse lecture, that will actually very easily combine these things. So the first issue you might notice is that when we look at our row names here, you've got this sort of dot number. You've got, you know, dot 11, dot 9, dot 10. And when you look here, you don't have that. Well, so how do you deal with that? Well, the easiest way is actually just to find these dots followed by numbers and remove them. So that's what we're actually going to do here in this section. So the first thing we do is we take our data frame, we actually convert these row names to a column. And so again, this is just sort of to recap, this is your tidyverse sort of material here. So if I take a look at that, I can see what it's doing. So basically it took those row names and it converted them to a column called row name. And that's what you see here. And now we're going to use a mutate line in order to clean this row name column so we can remove these here. And so basically, if you recall from before, this is the regular expression or regex pattern that is used to find a period followed by something. So this basically says find the period followed by whatever, and then replace it with nothing. So basically that means it's going to find this, replace it with nothing. It's going to find this, replace it with nothing. And then we're going to take all that and assign it to a new column called gene ID. So let's see what that does. Okay, so we can see that that's exactly what happened. We took each of our row names and then we basically just lopped off this version number at the end and then returned the cleaned gene ID. Now, we don't need this column anymore, so we can actually just select and remove it. And then finally, we want to do an inner join. So if you recall, an inner join will combine this data frame with this data frame based on a column, and then it'll make it so that the rows are only going to be kept if they're in both data frames. So it's only going to keep the rows where the gene ID was in both data frames. And if you don't remember that, go ahead and look at the resources for the workshop, and you'll find there's a really nice, and I'll actually just show it here. There's a really nice cheat sheet called the data wrangling cheat sheet. And I'll just pull this up just because it's kind of nice to see this, where they go through a lot of the aspects of data wrangling that are most common. And so, for example, you can see the nice description of how the inner join is working here. Anyways, so result of all that, we can see here that now we've got gene IDs and their corresponding gene symbols for all of our results. And so when we order by p-value, now we can see what are the genes that actually go along with this, which is great because, for example, if I know that there's some change in collagen that I'm interested in understanding, then it's really exciting to see, col 11A1 show up as like a super differentially expressed gene. So now we're getting into kind of the interpretation part. So let's take a look at the volcano plot and let's see if we can start to get an idea of what's going on here. Okay. All right, so now we've got our volcano plot, and one of the things you might notice is that because we did our log fold change shrink, it's actually a lot nicer than it would be otherwise. So if you plotted it without doing that, you would actually find the values are like way to each side and don't really make a whole lot of sense. So it's much easier to interpret the data visually now, which is why we do that. So we can see here the genes that have the greatest fold change and the greatest significance for differential expression, and then the least fold change, so the most underexpressed. So basically top overexpressed, top underexpressed genes. So how do we actually interpret this data? Well, if I go back to my original question, I was asking, what is the effect of EWS-FLI1 knockdown in Ewing sarcoma? And then how does that change cellular behavior? Have I actually answered that? Well, obviously not, or there wouldn't be anything left in this lecture. But why is that the case? Well, the issue is to actually understand how things are changing, I would need to go through one by one through each of my differentially expressed genes. I need to look it up. I need to make sure I understand what is that gene, how is it working, what processes is it associated with. So I might know off the top of my head, col 11A1 is related to collagen, but I don't know what TFPI2 is or MGP. So that would be the first thing. And then the second thing is I'm biased. So it's not really appropriate for me to try to visually inspect my data in order to draw some sort of meaning from it. It's better to use all the data in an unbiased way and then get a complete picture. So how do we do that? Well, the first way is overrepresentation analysis. And so what we're going to do is we're going to get our overexpressed genes and our underexpressed genes, and then we're going to go and do overrepresentation analysis to see what are the biological processes that are represented by these overexpressed and underexpressed genes. And that'll tell us how is the phenotype of the cell or the tissue actually changing under this treatment condition. So I take my results, I filter them, I say I want p-value less than 0.01, log 2 fold change greater than 2. It's a little bit conservative, but I think it's preferable to do that sometimes. And then I write that to a CSV file. So go ahead and do this with your overexpressed and then underexpressed genes. And so if you go back to the folder where you're currently working from, so for me that's just the downloads folder, I can see now I've got my overexpressed genes and my underexpressed genes in here. So let's go ahead and start with the overexpressed. I know it's probably a bit sacrilegious for me to open this up in Excel, but I actually think sometimes Excel can be a lot more convenient than R, particularly with stuff like this. So these are my overexpressed genes, and now I've got my gene symbols. So what I'm going to do is I'm going to take this list of overexpressed genes, about 1,400 long, copy that, and I'm going to actually come over here to my browser, and I'm going to use a web-based tool for enrichment analysis, and it's called enrichr. So if you just Google enrichr and you click on the first thing, that will be the right one. So actually, I'm gonna post this in the chat just in case anyone forgets where that is. So let me copy that again. And all you have to do is paste your gene list into the box here. So it tells me I've got 1,400 something genes. And then all I have to do is hit submit. So what this is doing is, oh, it's too fast for me. What this is doing is it's finding in all of these databases of gene sets, it's finding if any are overrepresented, significantly overrepresented in the gene list I gave it. And basically, that's just a kind of a quick and dirty way of figuring out what biological processes and other things are overrepresented in your overexpressed genes. So, for example, my treatment was to knock down EWS-FLI1. So if I come over here to these TF perturbations, so these are transcription factor perturbations followed by expression, and it comes from a particular database that they curated, I can see that I've got a ton of pathways here that are very significant that have to do with knocking down FLI1 and then seeing genes that are overexpressed as a result. And actually, if I look at this study, which I can do here, if I copy this and I paste it into Google, I can read about that study, and I can see that that study was actually done in a Ewing sarcoma cell line. So this is actually the same experimental design that I'm currently working from. So the fact that I see this enriched is a really good sign that tells me that my experimental design basically worked as expected. Does anybody have any, I guess, any questions about this part before I move on to the actual biological interpretation? Okay, so yeah, if you've got any questions, do feel free to ask in the chat. This part can be, I think we're going to go a little fast through this. This part can be kind of confusing at first, especially if you're already not super familiar with overrepresentation analysis, like what does this really mean? But hopefully it'll kind of become clear in a minute here. So I'm going to move from transcription factors because I don't really care about that. I'm going to move to pathways because that's more what I'm interested in learning about. What are the pathways that are changing when I knock down EWS-FLI1? So just looking at this, knowing that when you see something in red, that means it's significantly enriched. Can anyone take a guess as to what process, like what like in just a large scale view, like what process is being dysregulated here? Any cancer researchers? So Yi says cell cycle. Yeah, there's probably something going on with cell cycle. Although I would say that's probably not the thing that jumped out at me first from looking at these. So I guess I should ask in a broader sense, is there a theme here? If you're looking at all of these results, is there a theme? Yeah. So Subhash said extracellular matrix. Yeah, I would definitely agree with that. And Yi says metastasis. I wasn't gonna go there right away, but yeah. Yeah, so yeah, adhesion, metastasis, extracellular matrix, all of that. So especially if you're a cancer researcher, this should just jump out at you because when you're thinking about what happens in the worst case scenario with a cancer, well, it becomes drug resistant and it metastasizes, and then it's really hard to do anything about it. And one of the main processes involved in that is upregulation of ECM interactions and angiogenesis factors and focal adhesion. And even down here, if you scroll down a little bit, you can see epithelial mesenchymal transition. So this is all pointing at a very metastatic, very invasive phenotype, right? So let's now. So there's other things we can explore here, but we've already got kind of a good idea of what we wanted to learn. So let's go back to our original question. Our original question was this: what is the effect of knocking down EWS-FLI1 in Ewing sarcoma? How does it change cellular behavior? And then as an addendum to that, are drugs that do this, are drugs that knock down EWS-FLI1 a good idea? Well, judging by the results we've already seen, we could kind of make a case that maybe they're not the best idea. Because, if they kill the cancer and then, that's it, then that's great. But if they don't, then they've got a pretty good potential to cause the cancer to undergo some sort of invasive transformation, which is not what you want. All right, so just in a nutshell, that's my like, kind of shorthand biological interpretation here, just to give you a glimpse into how I would think about doing this. Before you do the first hands-on activity, does anybody have any questions about what I've done here, or kind of how I'm interpreting these results? Okay. So the first activity is to find a study and recount and basically do what we just did, to actually analyze it and see if you can come up with, at least at a surface level, a biological interpretation of either the overexpressed genes or the underexpressed genes. Just get an idea of what's changing and in which direction. So I'm going to give 20 minutes to do that, because I think that's just a really important activity. And we might not get to everything else this time. We might have to pick that up next time. That's all right, because I think the main meat of what we're doing here is definitely what we've already covered. So go ahead, take about 20 minutes. And if anyone feels comfortable at the end presenting what they've done, please let me know, and I'd be happy to have you do that. Okay, so hopefully everyone's had a chance to try their hand at that analysis. Does anybody feel brave enough to share what they did? If I give you another 2 or 3 minutes, will anyone share? Okay, Simon says maybe. So actually, Simon, what you're describing sounds like a good uh, thing to discuss, just because it sounds like you're running into an issue that can very well happen to everyone. Would you actually mind sharing that? Thanks.
Can I share screen?
Yeah, go for it.
That work? Everyone see code?
Yep.
Okay, cool. So I looked for things that had to do with Parkinson's disease because I looked for things that had to do with tauopathy, and there wasn't anything, and then I got sad, and so I looked for what I worked on last. So if we look at project info, I went for the one in the middle there. Come on.
Okay, great.
This guy right here, they looked at gene expression from 6 PD cases and 8 controls, which sounds nice. And so then I copy pasted your code and replaced some stuff.
Great.
And so the HO underscore is just it's the hands-on to keep the variable separate from your code.
Sure.
So if we look at the counts, where'd they go? There we go. That's what they look like.
Great.
And then we look at the column data. So they had 4 controls, 2 sporadic PDs, 4 non-PD, which I'm assuming is like motor disease or dementia, but not PD diagnosis. Okay. I'm not sure. And then they have 4 other PD cases that presumably are genetic if the others are specified as sporadic.
Sure.
So then I went with control PD, like this. And then made the DSeq object, transformed it into the Pca. And so this is what it looks like. And so I'm just like, something's off. So, I mean, this guy right here, this guy looks like some sort of outliery sample. But yeah, I'm not quite sure. How do I check that this vector here, so when I look at RC gene condition, like
sure. Well, run lines 121 and 122 again, because that's going to show you your updated column data.
Oh, so I'm just an idiot and didn't do that. Ha ha ha. Okay, no, that did work.
You did have it right.
Okay, good. Yeah, I was just like, why can I not find, like, I thought it was supposed to be in the column data now, but I just was an idiot and didn't run it again. Okay. Well, then I would honestly just put HOs at the end of everything in your code and see if I need to change other stuff, because that's how I did it to get it done quicker. I didn't like start from scratch and start typing, because that would have taken way longer.
So just out of curiosity, did you actually run the analysis?
Oh, yeah.
Okay. I was just curious because like the PCA, I mean, I'll tell you that PCA doesn't look good, but I was just curious if like you would end up with any significant genes, or like if it would just be like a really small number. Well, because like I've seen that before with patient data, especially, which it sounds like you have.
Yeah. Well, also like, look, there's the PD is split into sporadic and not. Now, if I had to bet money, I'd say these two up here are the sporadic ones. But who knows? And then these 4 might be the genetic ones, but let's see.
Yeah. I don't know. I don't know if Dr. Chen, if you want to weigh in, or Dr. Pertzenleidis, if you want to weigh in on this.
But I'm actually not exactly, since I haven't run this one, but why the group only show the control and PD, but it seems, oh, you convert the control PD, you didn't actually name the non-PD separately.
Correct. Yeah. So because their abstract talked about just 6 PD cases and 8 controls, I decided to group the sporadic PD and the PD together as the PD cases, and then the Cs and the non-PD together as the control cases.
Okay. Yeah. I won't be that actually surprised if it's a patient sample. So.
Yeah.
Yeah, I think it might be.
Although, isn't it iPSCs?
Yeah, let me see here.
Oh yeah, no, that's DE-seq. I feel like DE-seq sends that message about replacing outliers when it's like just not super happy.
Oh yeah, look at that.
Yeah, I don't know. I see that. I usually only see that when the differences are not very strong.
What?
But anyways, so yeah, that's really interesting. It's I think it's very instructive to see that, because like I remember the first time I saw a PCA plot like that with patient data, and I thought I had messed it up, and I probably lost an entire day trying to like figure out what I did wrong. And then I realized that at the end of the day, like when you're talking about patient data, you have so much variability person to person. This is not like genetically identical data anymore, like you get with mice and with cell lines.
Right.
So
great. So I would be the to be more. I think for the PCA, you probably want to use all the annotation, and after you've done the PCA, you may just do the design and putting the whatever necessary. So it's always good to see the raw data to begin with. Then if you want to remove the sporadic and also the non-PD, then you do that afterwards. So in a way that you can clearly see the difference. Yeah.
Okay, so since it sounds like you're probably not going to get a whole lot from that, did anyone else have an example where they were able to get all the way to enricher?
Ooh, yeah, that's ugly.
Yeah. Well, okay, so I can actually, I guess with our last 15 minutes, I can just go ahead and go on to showing you at least how to do this instead of doing it online in your web browser, how to actually do it in R. And then just briefly touch on gene set enrichment analysis, which in many respects is an improvement over representation analysis, which is what we're currently doing. So, okay. So let's do this instead in R, right? So the way we're going to do this is we're going to actually do our filtering again. So this is our filtering that we did before, but instead of writing it to a CSV file, we're actually just going to get the genes. So when we do pull in tidyverse, that's just a way of pulling out a column and turning it into a vector. So we're going to do this, and this is going to give us our vector of overexpressed genes. And we can, oops, that's not what I meant to do. We can see that here. So that's our 1400 something overexpressed genes. Now we're going to use the msigdbr function in order to get some gene set annotations. And so just to really briefly touch on this, msigdb comes from this site. Oops, that's not what I meant to do. It comes from this site. So this is GSEA. These are the people who make GSEA. And it's the Broad Institute and UCSD. But besides making GSEA, which is the technique you can use for biological interpretation, they also have a database of gene set annotations that you can use in order to do these analyses. So just like before we saw with Enrichr, you have all of these databases of gene sets, well, you actually have the ability to get those from here as well. And so they've got hallmark gene sets. That's a collection of gene sets that they curated themselves. Positional, so that's like chromosome 1, chromosome 2. And then they've got just other collections. So gene ontology, if you're familiar with that, that would be C5. And so if you ever need to know which collection contains what gene sets, you can always come here and read these descriptions, and then that'll give you a pretty good idea. They actually, I didn't even realize this, they just added this like within the last few months, this new collection of cell type signature gene sets. Definitely inspired by some single cell studies. So, all right. So what we're going to do is we're going to pull from this database. We're going to get, human, we want human genes, and we want C5. So if you recall, C5 is our gene ontology annotations. So we're going to pull that here. And that takes a second. That's actually quite a bit of data. It's a pretty large data set. But yeah, it shouldn't take too crazy long. Okay. This was so fast earlier. It's crazy. All right. I'm just also going to remove this because that can also slow things down. So we can see here there's about 1.2 million lines in this data frame and 17 variables. So if we look at the head of the data frame here, if we look at it to just get kind of an idea of what's going on, we can see it's not a data frame at all, it's a tibble. And we can see here the columns that are in here. So what you're basically seeing is that every line is a gene, and then there's a gene set that corresponds to it. And actually all we care about for right now is the gene set name and the gene symbol. So we're just going to select only those. So now let's take a look at this. So we can see here, I've got genes and I've got the gene set they correspond to. So this gene set, I don't know how to say that. This gene set though, it's these genes. It's, GDP, GP1, MTAP, PYGB, and so on and so forth. So this gene set comprises these genes. And now all we're going to do is we're going to take this information, we're going to take our gene sets, and we're going to take our overexpressed genes, and we're going to plug them into enrichr, which is a function from the clusterProfiler package, and go ahead and run that. That's going to actually do the overrepresentation analysis. It's a little slow, certainly not as fast as that web tool, but if you want to do it in R, this is a pretty good way to do it. There's also some advantages to doing it like this because you're actually going to be able to use some built-in plotting functions built into the clusterProfiler package. And all right, so that's done now. So the result is about 80 megabytes. It's kind of large. But all we really want to see right now is the data frame version of that result. So we're gonna run this. We're gonna take a look at the data frame. So let's go ahead and see what that shows us. So it gives a few columns. It gives the ID and the description, which are actually the same thing. It gives you the gene ratio, BG ratio. Don't worry about those for right now. What you're really going to want to focus on is your P adjusted value and your Q value. So there's really not, these are actually, it's kind of hard to know which one's more important to look at. I personally don't know. I think it's fine, honestly, whichever one you choose. But I would say probably more for the purposes of clusterProfiler, they probably prefer if you look at Q value. And it's already ordered by Q value, which is nice. So basically the top result is the first result, and it tells us what is the most overexpressed pathway enriched in our results. And that's because we gave it our overexpressed genes. So just like we saw before, we're seeing extracellular matrix, we're seeing substrate adhesion, we're seeing all the stuff we saw before. So just like I said before, it's the same thing as what you did in your web browser, except now you're doing it in R. And so what if we wanted to actually look at our results? Well, we can use some built-in functions such as dot plot, and that's actually going to show up here. So you might see this in published papers because actually a lot of people use this package when they're publishing bioinformatics papers. But this is just one way to see the top hits from your overrepresentation analysis, kind of nicely shown. So color is the p-value, size is the size of the gene, or size of the, yeah, I think size of the gene set, maybe. And then gene ratio is kind of a similar thing. Honestly, I don't really see any need to pay attention to the count or the gene ratio since it's the p-value that really matters, but people like to see this as well. Anyways, so that's overrepresentation analysis. Now, I would say that overrepresentation analysis is probably, despite being the most popular way to do biological interpretation, it's actually not ideal for quite a few reasons. And I'm just going to really quickly show you why I think that. So if we look at our enhanced volcano plot again, and we actually use our genuine p-value and fold change cutoffs, because remember, to say overexpressed or underexpressed, we said p is less than 0.01, so that's 0.01. And we said fold change has to be greater than 2 or less than -2. So let's take a look at that plot. And it's, I guess it just takes a second here. So this is what that actually looks like. Now, everything that's in red on this side, we called that an overexpressed gene. Everything that's in red on this side, we called that an underexpressed gene. And this leaves something to be desired because, for example, if I look right here, right between these two spots, now there's a world, in my way of seeing things, there's a world of difference between these two genes here. And basically, if you're on this side, you're overexpressed. If you're on this side, you're nothing. So is that really accurate? Well, obviously not, because these genes are right next to each other. There's no magical line where something becomes overexpressed or underexpressed. It's just arbitrary cutoffs that we assign. P-value of 0.05, it is an arbitrary cutoff. It's one that we've all agreed on, but it is at the end of the day arbitrary. So there's a question of like, is there a way for us to use this distribution, use the whole continuous distribution of fold changes or p-values and actually get a more sensitive analysis technique? And in fact, there is. That's actually the motivation behind gene set enrichment analysis. The other, actually, I should say there's one more reason. Overrepresentation analysis can suffer from p-value inflation. So if you have a larger number of so-called overexpressed genes, then you actually end up with disproportionately more significant gene overrepresentation analysis results. So that's actually another issue. It shouldn't be that having a larger number of genes gives you more significant results. So for those reasons, they were motivated to develop GSEA. So I'm not going to explain how GSEA works. If you take Dr. Chen's and Dr. Perturbations' class, I think they're going to go into a little bit more detail about how it actually is working there. But just so you know how to do it, I want to show you here. And then for homework and on your own, you can use this. So the first thing you're going to have to do is you're going to have to actually rank all of the genes in your results. And the way we rank them is not by fold change. Some people do it that way. That's really not a good way to do it because we've already seen fold change can be wildly inaccurate, especially when you have low count values. Typically, what you're recommended to do is to rank all of your genes by the negative log 10 of the p-adjusted value times the sign, so plus or minus, of the fold change. So let's see what that's actually doing here. Let's take a look at that. So I've just made this new, oh, I guess I didn't. Oh, ResDF2, right. I've just made this new column in ResDF2, and it's got my GSEA metric. So basically now you can see here, if I look at the top overexpressed, I can see, well, let's ignore this for right now. I can see here, 287, because that's the p-adjusted value. That's a negative log 10 of the p-adjusted value times the log fold change, which is positive, or the sign of the log fold change, which is positive. And if I look at the bottom, then I've got negative 282 because that's the log 10 of the p-adjusted times the sign, so -1 of the log fold change. So hopefully that kind of makes sense how that's working. Now the issue is that we've got infinities, and the reason is because, I mean, actually, just out of curiosity, does anybody know the reason for that? Anyone who's kind of a math or numbers nerd?
So you had p-adjusted values that were so small that they were considered 0 by R.
Yeah.
And then you try to take the log 10 of 0, which doesn't exist, but the limit of log x as x goes to 0 is infinity. So.
Exactly. Yeah, that's exactly right. So that's why we're getting this value here. And that's going to really mess things up. So why is this 0? Well, Simon actually just said the reason. It's because R has a lower limit for the smallest number it's willing to deal with, essentially. And that is around 1 times 10 to the negative 307th. That's about the smallest number it's willing to deal with. So you can actually find that number by using this dot machine double x min. That'll tell you the smallest number. So this is the smallest number. So what we're going to do is we're going to do a mutate where anytime the p adjusted value is 0, we're actually going to exchange it for that smallest number. And then we're going to calculate that metric again. So let's now take a look at our new results. Okay, good. So we can see instead of having infinity here, we've actually got real numbers. Now they're all the same, but they're still real. So that's nice. All right. The other thing we need to do is we need to remove any NAs, and we need to order these results in descending order. So let's now take a look at that. All right, good. So now I've got everything's in descending order. There's no NAs anywhere in here. So if I go to the bottom, I can see the most underexpressed, or sorry, the strongest underexpressed. And then if I go to the top, I can see the strongest overexpressed. And now I've got a ranking, which is good. So let's take a look at a histogram of that, just to give you an idea of what we're seeing here and what we're going to try to do. So this is a histogram of all those GSEA ranking values. And so we can see we've got negative 300 something over here, positive over here. And so we've got this really long-tailed kind of distribution on both sides, very long tails, and then big spike in the middle. And what we want to do is we want to take this rank distribution of genes, and we want to see if we can discover any gene sets that are overrepresented at the top of this list or the bottom. So the top being overrepresented in the overexpressed genes, bottom being the underexpressed. So to do that, the first thing we're going to do is we're going to actually get this as a vector. And so that's what I'm doing here. I'm saying, select these two columns, make them distinct. So that's basically so that there's no duplicates, just something it's good practice to make sure there's no duplicates in general. And then deframe is a way of taking these two columns and turning them into a named vector. So this is more tidyverse stuff. So let's take a look at that now. I'll take a look at the head of that ranks vector that I've just made. So we can see that it is a named vector. So our metric is the number, and then the name is the gene name. And so it goes all the way through. It's got all of our genes in it. So to actually run GSEA, all we have to do is use this GSEA function from clusterProfiler, give it the ranks, and then also give it our gene sets like we used before for the enrichр function. So we run this. It's a little slow. There's actually a more efficient implementation, but I think this is completely fine because this is kind of a little bit more user-friendly, I would say, than the other way of doing it. And so this is just going to run. It's going to see if it can discover if there are any gene sets overrepresented at either end of our distribution. And hopefully it doesn't run for too much longer. If anyone wants to know how the math here is working, the 2005 paper, you can find that. That's actually on that website where I was getting the MSigDB. That's on there. And if anyone is interested in learning about kind of the math behind this and behind other sort of similar methods like the KS, I don't know how to say their actual names, the KS tests, which are kind of a family of similar statistical approaches. I'm pretty sure Dr. Chen goes over that in his lectures.
Just to wait when you're waiting for your program, It's Kolmogorov, Smirnov, so you have to practice a little more.
Yeah. I just didn't want to, because I feel like I can get Smirnoff pretty easily. I feel like that's, as like an American, that one comes pretty easy. But Kolmogorov is definitely harder.
Well, you got the result anyway, so.
All right, so let's take a look at the results. So it looks like I had 413 significantly enriched gene sets, so that's great. And so the way this works is that rather than just showing you the p-value and the p-adjusted value, you actually have this normalized enrichment score now. I personally choose to focus on this because I find that this is actually a much better indicator of the degree of overexpression or overrepresentation or underrepresentation. So if I go and I look at if I sort it and look at the top hits by NES, I can see, again, I've got my ECM, my, collagen. So this is stuff we already knew about. But what if I looked at the underexpressed ones? Now I start to see some interesting things. Now, I didn't have time to go through this with you, but basically none of this came up when I did the overrepresentation analysis with enrichr. So that's really exciting to see this now here. So I see ribosomal subunit, catalytic spliceosome, RNA splicing, mitochondrial translation. So some really interesting stuff that I'd probably be interested in exploring further. But just for the sake of time, I'm going to show you the last part here, which is how do you now plot this. So you can use a dot plot, but you'll see if you do the dot plot, it's not super informative because it's only showing you p-values. It's not showing you the normalized enrichment score. And also, people don't typically represent gene set enrichment analysis results like this. So I'm going to show you how they do represent it. And I don't know what the actual name for this is, but they just call it a GSEA plot in clusterProfiler. So what is this showing us? Well, along the bottom, we have our ranked gene list. So it goes from 0, that's our top overexpressed gene, all the way to like 35,000, which is our top underexpressed gene. And we can see here in this ranked list, we can see where did this gene set fall? Where did the genes from this gene set fall? And we can see that they pretty much all fell right here at the beginning. And so what does that tell us? Well, that tells us that this Go cell matrix adhesion biological process, that is occurring in our overexpressed genes. That is much more enriched at the beginning. It's much more enriched in the group of overexpressed genes. And so without having to assign an arbitrary cutoff, I was able to determine that using gene set enrichment analysis. So again, this is much more sensitive. And statistically, it's I think people would argue it's probably more robust. So I can also look at ribosomal subunit, which I'm pretty sure is my top underexpressed. And so I can see here when I do that, yeah. So it's now the opposite. So this was my top underexpressed. So it's on the other side now. It's towards the end of my list. And I can see it's going now the opposite direction. All right. And then I've got some code here to show you how to make what is basically kind of a summary of the 4 top pathways. So I'm going to show you just really briefly what that ends up looking like. And then if you've got any questions about how did I do that, do feel free to ask or, come to office hours tomorrow, or just to show you kind of like what this outputs though. All right, so now I've got my figure showing my top 4 overexpressed gene sets. And I've even got little kind of figure labels like A, B, C, D. I don't know if you'd actually publish something that looks like this. It's probably more for lab meeting, but it's still kind of nice to be able to do that. And so, yeah, there you have it. This might be how I would present this, for example, in a lab meeting. And if I was willing to put in some more time to clean it up, even, possibly in a paper. All right, so does anybody have any questions before we break? Okay, well, so that pretty much does it. We'll probably review a little bit of that next time as well, but I would say for homework, go ahead and do the gene set enrichment analysis and the enricher in R with the data that you found. And if you weren't able to complete hands-on activity number 1, go ahead and make sure you get that done before next time, because next time we're going to talk about where do you actually go to get data when recount is not enough. All right.
Sorry,
So
just a quick question. So you do the enrichr and GSEA all from the cluster profile, right? You didn't using it's a native the GSEA version.
Oh, so are you talking about like doing the web app, or not the web app, like downloading the desktop version of GSEA?
They also distribute the GSEA also has its own R package. But I look at your R code. It looks like you only include the cluster profile, which also I think it has. I don't think I personally used it. So
I like to use the clusterProfiler one just because it makes the plots automatically, but there are many implementations of GSEA you can use in R, which is kind of nice. And actually, sorry, Simon just asked a really good question, which is there a reason we would ever do enricher when GSEA is available? So we're pretty much over time. So if you're not interested in hearing the answer to that, I'll see you next time. But I think this is a really good point to address. So I'm going to stick around. And if Dr. Chen and Dr. Perturbations, you'd be willing to stick around, I would really love to hear what you guys think on that. So I'll just say, for my part, I use enricher all the time for like a super quick look at what's going on in a differential gene expression results, sheet. And I'll use the web tool usually when I'm doing that, because you can just drag and drop your gene list right in there. And it's useful for times when you don't have the ability to supply a ranking metric. So ranking is really a prerequisite for gene set enrichment analysis. But for enricher, all you really need is just a list of genes that you want to analyze. That being said, I think anytime you're doing any sort of serious analysis where it needs to be more robust, GSEA is probably a better thing to do ultimately. But I don't know, what do you guys think? Dr. Chen, Dr. Perturbations, what do you think?
I pretty much agree, but for example, I just give the example, not necessarily the reasoning. If you have a ChIP-seq, you end up with a bunch of binding site near the genes, and you want to see what these genes function in. I guess you can only do the overrepresentation in this case. So that kind of made the enricher or any other overrepresentation method necessary in this case. Yeah. So there are utility for different purpose. That certainly is true, though.
Yeah. So Simon, your question, GSEA metric. Yeah. So like what Dr. Chen is saying is basically like if you do ChIP-seq, then you'll get a list of genes that were bound by like a transcription factor, but you're not going to have like a good p-adjusted value and log fold change to go with that. So you would pretty much only be able to use the list of genes itself without really having the ranking metric.
No, that makes sense. Yeah, I didn't think about a ChIP-seq scenario where it's just binding or not so much binding. But so for like a differential gene expression where an LFC is available, it would be more.
Simon, I also see another question you just posted there. It's a very good question. Actually, we actually we talked to my programmer just today about this one. So I think it's certainly you can use p-value. You can also use for HR or DC2. You can use the statistic that generates. You can use all these things that already carry the sign, but either way though, it should work. But you can just forego the sign as well. Just go with the p-value itself if you want to. It doesn't really need to take a log because it's rank based though. So just for the sake that if you want to look at the functions group, bunch of genes that group together, whether you rank them by p-value or rank them by full change statistics or anything. Yeah, either way, depends on the gene set, though. It's pretty much that way.
And so Dr. Pertzamlia says, doesn't enricher let you look at things in parallel, meaning several sets at the same time? So are you talking about, just so I make sure I understand, like, when I'm looking at it like this, I can see all these different databases kind of at the same time? Is that kind of what you mean?
Much.
Oh, sorry, I didn't hear what you said.
That was a yes.
Okay.
Okay.
Yeah, that's one of the things I do really like about it as well. Now, I think that what they're doing here is actually something that is totally achievable with gene set enrichment analysis, but for some reason, no one's done it yet. So for right now, yeah, I think they're the only ones where you get this really nice, like, side-by-side database by database look at it. So he asked a really good question, which I actually don't know the answer to. Dr. Chen or Dr. Pertzamlia, do you know the answer to that?
It is what this rank method. So the oh, you mean the GSEA? No, GSEA doesn't really care how you rank the data, the input data. So you have your own variation. So I mean it depends on which software you use for the GSEA. So that actually have a long story by itself for the GSEA. Either the standalone or the programming side. So it's getting a little lengthy to describe. So I would be probably, you may want to talk to Henry or maybe me at a different time. We can discuss more detail. So.
Okay, so thank you all so much. And yeah, I'll see you all next time.
That's good. Thank you, Henry. It's always learn something new when other people talking.
For sure, for sure.
Thank you.
Good job, Henry.